So today we are going to discuss about production of penicillin. The production is mainly by fermentation method. So penicillin, it was the first antibiotic discovered, and it is one of the most important milestone in the medical history, the discovery. And it was discovered in 1928 by Alexander Fleming, a bacteriologist, at St. Mary's Hospital, London. So while Fleming was working with some Staphylococcus aureus bacteria, that means he was culturing Staphylococcus aureus bacteria in in his petri dish. In some areas, he failed to find the growth of Staphylococcus aureus. That means in the petri plate, he was culturing Staphylococcus aureus, and in some areas, the bacteria, that is Staphylococcus aureus bacteria, is not growing. Okay, then he checked that area. Since that vicinity of that area, he could find that there is some other thing that is growing in that area. Okay, so that area, then he identified that that area is mainly occupied by another fungi or another mold. Okay, and this fungi entered into his culture plate accidentally. It is entered as a contaminant. That means Fleming, he was culturing Staphylococcus aureus bacteria in a petri plate, and while culturing, in some areas there is no growth of aureus bacteria, and those area that didn't have the growth of aureus bacteria, he checked why, what is the reason why in some area this bacteria is not growing. Then he found out that in those areas there is the growth of another fungi. Okay, there is the growth of another organism in those area. That organism is killing the aureus bacteria. Okay, and this fungi or this mold was later identified as Penicillium notatum, and this Penicillium notatum bacteria is secreting some antibacterial substances, and these substances was killing the Staphylococcus aureus bacteria. Okay, so Penicillin notatum, it is a fungi or it is a mold. This fungi will secrete some antibacterial substances into the medium. So due to these antibacterial substances, the Staphylococcus aureus bacteria couldn't grow. Okay, so finally he named that bacterial substances as penicillin, and that in that way this penicillin became the first naturally derived antibiotic for treating bacterial infections. So we know that most of the bacterial infections are mainly due to the bacteria such as Staphylococcus aureus, Streptococcus, E. coli, etc. So all these bacteria can be killed by another microorganism. It is a fungi, that is Penicillium notatum. This Penicillium notatum will secrete some antibacterial substances called penicillin. So with this help of this penicillin, we can kill the bacteria. Okay, and by chemically, these penicillins are belong to beta-lactam antibiotics. Since if you are looking the structure of penicillin, it is having a beta-lactam ring. Then coming to the mechanism of action, so we said that this penicillin is having a beta-lactam ring. So this beta-lactam nucleus is the thing that is producing the antibacterial activity, because it will inhibit the bacterial cell wall synthesis by blocking the enzyme transpeptidase. So transpeptidase is the enzyme which is required for the production of cell wall in bacteria. So this penicillin, it will inhibit the enzyme transpeptidase in the bacterial cell wall and leading to the cell wall lysis in the bacteria. And for penicillin was the naturally occurring antibiotic, and nowadays modifications in the side chain produces many semi-synthetic and synthetic derivatives of different penicillin drug. Okay, the natural source of penicillin is the mold Penicillium chrysogenum. So earlier it was known as Penicillium notatum. So later it is called as Penicillium chrysogenum, which is cultivated under aerobic fermentation conditions. So in order to produce penicillin, what we are doing now in large scale, for the large scale production of this penicillin, we are culturing penicillin in certain medias by providing some cultural media and aerobic fermentation. We can produce penicillin in larger amounts. Coming to the production, so penicillin notatum was used earlier. Nowadays, I said that Penicillium chrysogenum is the organism which is used for production of penicillin. Among this Penicillium chrysogenum, the Q strain, that is the Q176 strain, is the strain we are using. So this is the picture of Penicillium chrysogenum mold or the fungus, and the Q strain 176 strain is used because this strain is having high yield capacity. Okay. Then coming to the production. So before the fermentation process, the first thing that we required is the inoculum. Okay, inoculum is nothing but the microorganism, the fungi. Okay, so inoculum preparation, for the inoculum preparations, we are growing the penicillium fungi in a cultural media. Okay, so for that, at the first step, the lyophilized spores of growing hyphae are suspended in water, or sometimes we can add some wetting agents and all, such as sodium lauryl sulfate. Then they are added to the flask or bottles containing the cultural media. Cultural media will be wheat bran and some other nutrients, and they are incubated for 5 to 7 days at 25 degrees Celsius. And after some times, what happens? The sporulation will take place, and this entire process is repeated several times. And finally, we will get an inoculum. This inoculum will be at a concentration of 5 into 10 raised to 3 spores per ml. Okay, so per ml, this much concentration of the spore should be there, and that spore is we are adding into the fermenter. So in order to, for the fermentation process, we will be requiring a fermenter. Into this fermenter, we are adding the inoculum. Inoculum is nothing but the microorganism itself. Here, the microorganism we are using is the Penicillium chrysogenum. So this Penicillium chrysogenum organisms, first of all, we are incubating into a cultural media for 5 to 7 days at 24 degrees Celsius. So we'll get a heavy population of this bacteria. So it is not the bacteria, the fungus. And after that, this fungus we are adding into the ferment. Then, so we said that in order to inoculate, or in order for the cultivation of this microorganism in the fermenter, we require some cultural media. Okay, so the components of the media mainly includes carbohydrates such as corn steep liquor, lactose, nitrogen source such as soy meal, yeast extract, whey, calcium carbonate, potassium dihydrogen phosphate, vinyl acetic acids, magnesium sulfates, anti-foaming agents, etc. So first of all, inside the fermenter, we are placing the petri dish or something like that. Into it, we are adding this media. Into this media, the inoculum that is prepared in the other slide. Okay, the inoculum that we have prepared, that is 5 into 10 raised to 3 per ml of spores, will be added into this cultural media in the fermenter. Then before starting the process, the fermenter should be sterilized with the superheated steam. And the production process, it is happening in 2 steps. It includes the growth phase and the production of penicillin phase. So in the growth phase, it is the initial vegetative growth phase where the spores are allowed to grow for about 40 hours. So the inoculum we are added in the fermenter. This fermenter is having some cultural media. So for 40 hours, we are allowing the spores to grow, and after 40 hours, what happens? Within the 40 hours, the cells will be increased up to double the amount. Okay, so the inoculum, if it is 5 to 10 raised to 3, after 40 hours, it will be doubled. Then after 40 hours, next phase is the production phase. Okay, so in between, what happens is that after 40 hours, the penicillins will form a filamentous broth. So the viscosity of the media may increase. So when the viscosity increases, it will hinder the oxygen transfer. Therefore, the fermenter should always contain an ample supply of oxygen because penicillium is an aerobic organism. We already said that for the production of penicillin, which type of fermentation we are using? Aerobic fermentation. Why? Because penicillin is an aerobic organism. So, after the production of penicillin, when the penicillin started growing, what happens? The viscosity of the medium may increase. This will hinder the oxygen supply. Therefore, we should provide oxygen from outside into the fermenter. And the pH also should be maintained at 6.5. So after 40 hours, so the 40 hours is the growth phase. The spores or the fungi will be multiplied in double. After 40 hours, what happens? The penicillin started to secrete. So from the Penicillium chrysogenum organism, the penicillin is the antibacterial substances that will kill the microorganism. Okay, so this penicillin starts to secrete. When the production of penicillin is very high, the growth of the culture will declines. Okay, so in order to increase the production of penicillin, we can add different components of the cultural media, especially the carbon source. Okay. Then during this time, some chemical changes will happen in the fermenter. Okay, that is especially in the there will be changes in the pH. Okay, at the initial stage there will be no change in the pH, but after some time, that means after 40 hours and all, the pH increases up to 7. Okay, 7 to 7.5. This is mainly due to the ammonia gas released from the corn-steep liquor. Okay, the corn-steep liquor will release ammonia gas. Due to this ammonia gas only, the pH of the medium will increases to 7.5. The temperature should be maintained between 20 to 25 degrees Celsius. And once the pH reaches up to 8, the fermentation process stop. Therefore, the pH always should be maintained at 7 by adding calcium carbonate to the medium. Okay, so initially the pH was 6.5. After 40 hours, what happens? The pH will rises up to 7.5. And if it goes beyond 7.5, that means if it is reached up to 8, the fermentation stop. So in order to avoid that, we should maintain the pH at 7 by adding calcium carbonate, and the oxygen level should be maintained at 25 to 60 millimole per liter of hour. Then after 40 hours, the penicillin begins to secrete by the fungus, and the entire process will continue for 6 to 8 days, and at the end of the fermentation, penicillin is extracted. Once the penicillin is secreted by the organism, next step is isolation and purification. Okay, so this penicillin is usually present inside it will be inside the mycelium of the fungus. So the mycelium and the cells are removed by fermentation, by filtration and centrifugation process. And nowadays we can use rotary vacuum filters also. Once the mycelium is removed, the penicillin is recovered by the process of solvent extraction. Okay, that means into the mycelium we will be adding some solvents, and by solvent extraction we are extracting the penicillin from the mycelium. So in order to begin this process, first of all, we are reducing the pH to 2 to 2.5 by addition of sulfuric acid or phosphoric acid. Then following, we are adding amyl acetate, butyl acetate extra for the extraction of penicillin. Okay, the temperature also should be maintained at 0 to 3 degrees Celsius, because in higher temperature, the penicillin may get damaged. Okay, and the impurities can also be removed by treatment with the activated charcoal. And in order to precipitate the penicillin, we can use sodium or potassium acetate. Okay. Then for the further purification, we can use distillation, sublimation, column chromatography, etc. And any additional impurities can be removed by mixing the penicillin crystals with the volatile solvents such as ethanol, isopropanol, etc. Okay. And after the purification, the penicillin crystals are collected and dried under required pressure. So this is the diagrammatic representation. So first of all, inside the fermenter, we are adding the culture media. Into this culture media, we are adding the inoculum. And first phase is the growth phase. The growth phase is up to 40 hours. After 40 hours, what happens? The production phase. In the production phase, the penicillin chrysogenum microorganism or the fungi starts to secrete penicillin, and the pH may also start to increase up to 7.5. So when it is gone beyond 8, the production process will be decreased. So in order to avoid that, we will be adding calcium carbonate, etc. And after 6 to 7 days, what happens? The penicillin will be completely produced, and after 7 to 8 days, we will be extracting the penicillin from the mycelium cells by the means of different extraction process such as filtration, column chromatography, chemical precipitation, etc. Then finally, we are recrystallizing and drying at a reduced pressure. Okay, this is the production process. Coming to the uses we know that it is therapeutically used in the treatment of infectious diseases, such especially gram-positive bacteria. They will inhibit the cell wall synthesis, and commercially produced natural penicillins are penicillin G. They are used as human medicine 38%, and 12% is used as veterinary medicine, and 43% is used for the production of semi-synthetic penicillins. So there's not that much only about the production of penicillins. Thank you.